Good afternoon to all of you, and thank you for taking the time to join our seminar. I'm very excited to be here today. My name is María José Roca Cervera, and I work as a geneticist at Reference Laboratory Genetics. I'm sure that all of us have worked on cases where extra effort were needed to confirm the clinical diagnosis of the patient. In today's webinar, I'm going to present, by showing 4 complex cases examples, how our experience in clinical interpretation combined with the most advanced technologies and bioinformatic tools can end the diagnosis odyssey, solving even the most difficult cases. In addition, we will look at the importance of clinical information and the use of HPO terminology. I guess that most of you already know us, but for those who don't yet know us, I would like to tell you a little bit our laboratory. We are based in l'Hospitalet de Llobregat, Barcelona. Since our foundation in 1975, we have established ourselves as one of the leading laboratories in Europe for spatial analysis. In fact, we are the largest laboratory in Europe, 15,000 square meters, collaborating with over 400 hospitals and over 1,000 laboratories worldwide. Our difference is that we have a complete service, both for genetics and other special analysis. Our passion is to develop new diagnosis tools that have a significant impact on the quality of life of patients and their families, always prioritizing the interest and well-being of them. And let it be clear that I'm talking about patients, not customers. To guarantee this well-being, first, we offer quality diagnosis tests being approved by different accreditation bodies, including ISO 15189. Then, we try to diagnose the patient as soon as possible, although we are cautious in saying that we do it less than 30 days. Should any complication arise in the diagnosis process, in 95% of cases, we diagnose the patient and deliver the report in less than 15 days. And finally, we provide bioinformatics support through our new Redless Cloud and database. In case 3, we're connecting dots. We have detected a variant in a nucleotide and a deletion of 2 exons as the cause of the pathology in a patient with clinical symptomatology associated with the delayed connective development. In case 3, we're connecting dots. We have detected a variant in a nucleotide and a deletion of 2 exons as the cause of the pathology in a patient with a clinical symptomatology associated with delayed. In case 3, we're connecting dots. We have detected a variant in a nucleotide and a deletion of 2 exons as the cause of the pathology in a patient with a clinical symptomatology associated with delayed cognitive development. Then, according to the clinic they are referred to, and which we will later observe in detail, the most appropriate studies of our NGS service for these patients were clinical and whole exome studies. We have performed the clinical exome on those patients with a well-established phenotype, but suggestive of a wide group heterogeneous pathologies. We also recommend it for cases with a little clinical information of variable splicenet or not well characterized prenatal studies. In these patients, we sequenced 5,227 genes. We have performed whole exome on those patients with a complex phenotype with the need to detect new target genes or with previous inconclusive studies. Among these, we sequenced more than 20,000 genes. In both cases, we analyzed all the deleterious and pathogenic variants over the sequenced genes and the variants and known significance of the select genes using HPO terms. You may know an HPO term is a numerical term that includes all the genes associated with a phenotype. An example of this number, HP00012759. Within this numerical term are the 2,311 genes associated with the neurodevelopment abnormality to date. In the Human Phenotype Ontology database, the different phenotypes that exist are associated with the numerical terms that include the genes. There are HPOs that encompass symptoms with general involvement, such as abnormality in neurodevelopment, and more specifics, HPOs such as intellectual disability or global development delay. Hence, the importance of clinical input. As we study genes associated with a phenotype, or in other words, with a patient's clinical symptomatology, the contribution of a complete and specific clinical report is very important, given that, according to our experience, we have observed that the greater the clinical contribution, the greater the probability of diagnosis. Once we have agreed in which study was to be carried out on each patient, we proceed to extract the DNA sample in order to sequence and analyze it. Our own database, RevLifeDB, allowed us to rule out a large number of non-causal variants in the patient's clinic, reducing the number of variants boosts significance. This meant that we were able to produce complete and conclusive reports of results, providing personal genetic advice. So, once we have seen what type of studies we carry out of these patients and how the diagnostic process was carried out, let's see what findings there were in case 1. We had an 8-year-old girl who presented intellectual disability with features of autism spectrum disorder, psychomotor retardation, and facial dysmorphia with a tendency to cyanophrenia, continuous implantation of the eyebrows. Previously, genetic studies have been carried out on her, which are usually started in patients with intellectual disability, X-fragile, karyotype, and array CG8, all of them being negative or inconclusive. Celiac disease studies were also carried out with negative results. Therefore, whole exome was performed from the genes obtained from the different HPOs in the patient clinical symptomatology. According to the phenotype, phase 2 gene, a tool that prioritizes possible disorders on the clinical symptomatology and that we use in complex cases as a first contact, like other similar applications such as GARD or Poisson-Webb, reported as possible pathologies in the patient the Coffin-Siris syndrome, the KVG, the Cliffstrom, or other known MIMUs. When we perform the exome study, however, we detect the presence of a clearly pathogenic variant in ACN2A gene. It was a missense type variant, not reported in population frequency databases and reported clearly as pathogenic in clinical databases and scientific literature. The SCNH2 gene had been associated with seizures, ataxia, and development and epileptic encephalopathy 11, all with autosomal dominant inheritance pattern. If we compare the clinical symptomatology of our patient presented with the one described in patients with epileptic and development encephalopathy, we can see how the affectation of intellectual disability on autistic features coincide. However, it was reported that affected patients had refractory seizures. Epileptic seizures were so frequent that they limited the patient's ability to live fully and appeared in childhood, so it did not fit with our patient's clinical symptomatology. with the father, and so that variants in this gene, also not clearly pathogenic, have been detected in 5 patients without seizures, 2 patients with intellectual disability, 3 patients with autism, and 1 patient with 2 pathologies. So we wonder, are the dysmorphic traits associated with this gene, or are these other genes involved? For this, better research studies on the variant will be necessary. In spite of knowing the pathogenicity of the variant, in view of the findings of these new associations, we decided to carry out the study of the variant in the parents with the aim of confirming the suspicion of de novo origin. De novo origin was confirmed, supporting the causality of the variant in the patient's clinical symptomatology and increasing the phenotype spectrum to which the SCN2A gene is associated. In second case, we discovered a variant in a research gene, not previously reported as the cause of the pathology, placing this gene as a target in neurodevelopment disorders. We had a 7-year-old boy who presented neurodevelopment disorder, facial dysmorphia, camptodactyly, limitation of the extension and deformity of the interphalangeal joint of the finger, and then undescent testicle, cryptorchidism. We also had vision problems. Previously, he had also undergone genetic studies, X-fragile, karyotype, and RADGH, all which were negative. In this case, they referred that they had no relevant family history. Therefore, a whole exome was performed from the genes obtained from the different HPOs in the patient's clinic. According to the phenotype, phase 2 gene reported as a possible pathology of the patient the Wiedemann-Kolb syndrome, deletion of the chromosome 1p36, Coffin-Siri syndrome, or Cruduchat, among others. When we perform the exome study, we detect the presence of 4 variants of unknown clinical significance. One of them in SOX11 gene, associated with Coffin-Siri syndrome as predicted by phase 2 gene. Another in EP300 gene, associated with Menkes-Hillman 2 and Rubinstein-Tabby 2 syndromes. This was in the regulatory region. We also studied variants in these regions and synonymous variants since we have had cases in which they were causal. And finally, 2 variants in the PHEF12 and SEC24A genes not previously reported to any pathology, research genes. If we look at the clinical symptomatology that our patients present in comparison with the pathologies to which the detected variants are associated, we can see that it could be compatible with Menkes-Hillman syndrome, characterized by development delay, growth retardation, craniofacial dysmorphia, and autistic behavior. As with Rubinstein-Tabby syndrome, a malformative syndrome also characterized by dysmorphia and neurodevelopment disorder. As with major features of Coffin-Siri syndrome, a clinical and genetically heterogeneous disorder. The PHF12 and C24 genes were not among the phenotypically associated genes currently described. However, because of type of variant predicted by the nucleotide variant, we considered it interesting to pay attention to them and delve further. None had been previously reported in the databases. the variant of PHF12 gene was a non-step change in which the substitution of amino acid glutamine by a premature stop codon at position 687 was predicted, resulting in a truncated protein. The native protein had 1004 amino acids. The variant of the SEC24A gene was a splice substitution type change, in which splicing predictors estimate involvement. The pathogenicity of this variant could have been verified by RNA studies, but we decided first to try to rule out with segregation. In AGMD, two variants have been reported in PHF11 gene 12, one in a patient with development disorder and the other in a patient with pulmonary arterial hypertension, and three variants in the SEC24 gene in patients with autism. We decided to go to genome ID to see the probability of intolerance of genes to loss of function variants. One being totally intolerant and 0 totally tolerant, we saw us PHF12 gene was more intolerant to loss of function variants than SEC24 gene. This data support a possible pathogenicity of the truncated variant in the PHF12 gene. Additionally, we went to see expression studies, seeing how the PHF12 gene was expressed in central nervous system tissues, while the SEC24 gene was expressed in other types of organs such as the liver, lungs, and pancreas, therefore also supporting a possible pathogenicity of the variant in the PHF12 gene. In this way, we carry out studies of the segregation of the four variants, with three of them being inherited. One, SOX11 by father, and two, EP300 and SEC24A by the mother. Due to the fact that these genes was autosomal dominant, the inheritance pattern, or unknown in the case of SEC24A gene, and that the parents were asymptomatic, we allowed this variant as causal in the patient's clinic. The variant in the PHF12 had de novo origin, supporting causality of this variant in the patient's clinic and taking strength of the PHF12 gene as a target gene in neurodevelopment disorder. Once finished, we continue with case 3. In case 3, we connecting dots. We have detected a variant in a nucleotide and deletion of 2 exons as the cause of pathology in a patient with a clinical symptomatology associated with delayed cognitive development. We had a 13-year-old girl who presents neurodevelopmental disorder, hearing loss, obesity, and visual impairment with severe night blindness. No other genetic studies would have been done previously. A clinical exome was performed from the genes obtained from the different HPOs in the patient's clinical symptomatology. According to the phenotype, first two genes reported as possible pathologies of the patient include, among others, the syndrome of Bardet-Biedl, Rubinstein-Taybi, Mayer-Gorlin, and Carpenter. And a probably pathogenic variant with a 50% allelic fraction was detected in the BBS9 gene. A fraction variant not reported previously in any database or scientific literature. This variant encompassed the splicing acceptor site, so there was also the possibility that it was affected. Currently, this variant would have been classified as pathogenic, according to the updated Association for Clinical Genomic Sciences variant classification criteria, since the variants of loss of function reported distally. However, At the time of diagnosis, according to the American College of Medical Genetics and Genomics, ACMG criteria at the moment, it was considered probably pathogenic. This gene was associated with Bardet-Biedl syndrome, a rare hereditary multisystemic disease characterized by polydactyly, intellectual disability, obesity, underdevelopment of the reproductive organs, deafness, and retinitis pigmentosa. So it could fit with the clinical diagnosis of suspicion. However, it had an autosomal recessive inheritance pattern. So that leads us to the question, is there another variant? We saw the importance of Ler's deletion application in the BBS9 gene, seeing that although it was not very frequent, there were some isolated cases in which this type of variant had been reported. Therefore, we proceed to perform the CMB study in this gene by detecting a deletion of 2 exons, a variant of uncertain clinical significance. We had a variant with loss of function, a deletion of 2 exons in a gene compatible with the clinical diagnosis of suspicion. We therefore perform segregation, confirming the transconfiguration of the variants, each variant being inherited from a parent, and the sister being asymptomatic, carrying the deletion of 2 exons. The couple could have a 25% risk of having offspring effect in the future pregnancies. The added value of this case is that, as you know, various technologies were needed to detect CMBs of different sizes. Until now, to detect exon-to-exon or gene-to-gene size, it was necessary to carry out the study using MLPA. But there were many genes for which there was no proof, and that we couldn't study. Through NGS, with a single technology, we can detect CMBs from exon to full chromosome, becoming a cost-efficient method that improves diagnostics performance. And this is important, given that in neurology, 35% of the variants are CMBs, and more specifically, in rare pediatric diseases, 7.7%. And finally, we are going to explain the case 4. We have solved an enigma. We have detected an inherited variant in X-linked gene. And due to the inactivation of the X chromosome and the variety of the phenotypic spectrum, is not clinical causal in the progenitor but in her offspring. We had a male fetus with corpus callosum agenesis and ventriculomegaly in which legal termination of pregnancy had been performed. The parents were asymptomatic and had undergone another previous abortion with female fetus in the same clinic. Due to the scarce clinical data reported, in order to have a more precise diagnosis, a trio exome was performed, that is, the patient was studied together with his parents with the aim of directly ruling out variants in complete penetrant genes that the parents were carriers. The minor mean coverage and percent of risk to 20x in the patient was due to the fact that it was a paraffinized abortive sample. The HPOs used were on the slide, these ones. Thus, we detect a probable pathogenic variant in LA1 gene, hemizygosis in the patient, and heterozygosis in the progenitor. This was a variant that affected 2 functional domains, had not been previously reported, and all predictors estimated a pathogenic effect. This variant was at first of unknown abuse, variant. But since other variants have been reported in the same position as pathogenic, we decided to go for and it classified as probably pathogenic. In the patient, as seen in the AGB, it was detected with an allelic fraction of 87%. So it was validated by Sanhar, confirming its imagogosity. The gene had been associated with chromosome agenesis and several syndromes such as MASA or crash that include this clinical symptomatology with a recessive X-linked pattern inheritance. Ventriculomegaly had not been associated with this gene, so it's possible to think that it could be due to a new phenotypic association or that it could be due to another cause. Therefore, we had a variant in a gene that matched the clinical symptomatology, and when it was linked to X, it showed that the mother was not affected as a carrier in the heterozygosis, and the male fetus was affected as a carrier in hemizygosis. So we went to test the variant in the second female fetus to see that we could find there. In such a way, we saw that the fetus was a carrier in heterozygosis, which made the scheme unbalanced, since supposedly it could not be that the heterozygosis progenitor was an effect and that the heterozygosis female fetus was affected. But we trust our instincts and went further, investigating and seeing how in pathogenic variants in LA1CM gene, specifically in the case of MASA disease, variability in phenotypic expression has been described in heterozygous women as a consequence of the inactivation of X chromosome. This all began to make sense, so we decided to study the family of the parent. We had no sample available from the father, but we did have one from the mother and her sister, and they turned out not to be carriers. So, while waiting for the inactivation study, everything supports the causality of the variant. And once again, I like to highlight the importance of triexome. This type of study, accompanied by the complete clinical contribution, produces a very high increase in the possibility of findings a diagnosis. So whenever possible, we recommend that it be carried out. To conclude, we take home. Trios are better than a singles. So people, please, open-minded. Oh, I mean new association, right? We must do it with love. Look further to find the cause. And be careful with CMVs. The symptomatology is essential for diagnosis. And be careful too with biological processes such as X inactivation, which can lead to erroneous co-segregations. Solving this case requires a lot of teamwork, and at reference, this is what we pride ourselves on. So we would like to thank the whole medical and multidisciplinary team of the different centers and hospitals with which work, because with their support, we can achieve these innovations. We also wanted to thank and support all the people who are going through or have gone through a difficult situation because of the COVID. And thank you very much to all of you who are listening to us. It has been a real pleasure to discuss these cases with you, and we remain at your disposal to resolve any doubts you may have through our webpage or email too. So now I will wait 2 minutes to give some time to write down some questions. Okay, I will now select some questions that you have sent to us. If you have any additional question, please send them to webinars.referencelaboratory.is. Let's start with the first question from Peter. It's a good question. Parental segregation studies are conducted and the candidate variant is confirmed. Is the variant classified again? Yes. The variant is reclassified according to ACMG criteria with the new data obtained. And if this variant was described in a report issued for the previous patient as well with a new classification of the variant. Another question from David. I have seen that you sequence paraffin samples. This type of sample decreases the probability to find diagnosis? No, David. Fortunately, NGS technology and pre-analytical methods have evolved and are very advanced. At our laboratory, we have a lot of experience processing paraffin samples, obtaining high quality of DNA. In the worst case, at 20x, it's at 96%, for example, but at 10x, it's at 98%. With 10 reads, you can still catch most variants. Let's go to another question. This one from Claudia. In negative cases of clinical exome sequencing, do you advise to proceed with CMV analysis, or would it be more advisable to apply for a whole exome sequencing? We will first proceed to see if there are any CMVs of interest in the genes we have sequenced. Firstly, because they are genes that have been associated with the clinic that our patient presents. So we have to be sure and rule out all types of variants that may be present. And secondly, because NGS analysis of CMVs is very affordable. So by MLPA, we have a gene. By NGS, we have almost the whole exome. So we have to take advantage of that. Once we have rolled out of all the possibilities at clinical exome sequencing, we'll continue with whole exome sequencing to look for new target genes. Okay, at this point, I'm very sorry, but we don't have more time to proceed with the questions. As I said before, please send any additional question to webinars@referencelaboratory.is. I'll be very happy to personally answer all of them. It has been my pleasure to be here today, and I hope that you have a wonderful afternoon.